Welcome to the San Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Please like this video and subscribe. It truly helps support the San Science Experience. Today, we're diving into the really remarkable world of lysosomes. These are your cell's crucial recycling centers, absolutely vital for how cells function, their metabolism, everything.
Right, and here's the thing, the core challenge, really. Despite how important they are, lysosomes are under constant threat. Think about various things, pathogens, nanoparticles, even some drugs. They can all compromise that delicate lysosomal membrane.
And that's bad news, right? If it breaks.
Exactly. When that integrity is lost, you get leakage. Damaging stuff like digestive enzymes, charged molecules, they spill out into the main part of the cell, the cytosol, and that can trigger all sorts of problems, cell death, unwanted immune responses.
Yeah. Okay, so our mission today for this deep dive is to unpack what's new. We're looking at a review article, Lysosomal Membrane Homeostasis and its Importance in Physiology and Disease. We want to explore how cells actually sense this damage, how they repair it, or, get rid of the broken ones and make new ones. It sounds like a whole defense system.
It really is a multi-layered defense system.
It's just amazing how fast cells have to react. So what's the very first thing, the immediate frontline response?
Well, it's pretty impressive. The cell has these really rapid repair mechanisms. One key player is the ESCRT machinery. It's highly conserved, found in lots of organisms.
ESCRT, like a molecular patching kit.
Kind of, yeah. That's a good way to put it. It gets recruited super quickly to the damage site, and it works to seal the membrane. Often, it seems to do this by making the damaged bit bud inwards, forming these little vesicles inside the lysosome. And calcium ions, which kind of flood out when there's a rupture, seem to be a really crucial signal to get ESCRT there.
Okay, calcium sounds the alarm, ESCRT patches the hole. What else?
Then there's this other quick response involving the ER, the endoplasmic reticulum. The ER mobilizes, basically acting as a supply depot for lipids, like cholesterol. Specific proteins ferry these lipids over to the damaged lysosome.
And why lipids?
It makes the membrane more rigid, sort of shores it up, makes it less likely to get damaged further, so it's repair and prevention.
Interesting. Any other quick fixes?
Yeah, there's a third pathway involving an enzyme called sphingomyelin, or SMSA. This enzyme changes a specific lipid, sphingomyelin, into ceramide right there on the damaged membrane. And this change also encourages that inward budding of the lesion. It's another way to seal the breach, apparently independent of ESCRT.
Wow, okay. Multiple rapid repair options. But I guess sometimes the damage is just too much, right? Like beyond patching.
Exactly. Sometimes repair isn't feasible. The damage is too extensive.
So then what? A more radical solution?
That's when lysophagy comes into play. You can think of it as the cell's demolition crew.
Lysophagy. Sounds like lysosome eating.
Pretty much. It's a specialized form of autophagy. When a lysosome is severely damaged beyond repair, the cell tags it for destruction. A new membrane, called a phagophore, wraps around the damaged lysosome, forming what's called an autophagosome. Then, this structure fuses with a healthy, intact lysosome, and everything inside gets degraded.
How does the cell know which ones are too far gone?
Good question. There are signals. Things called galectins can detect sugars that are normally inside the lysosome but get exposed when it's damaged. Also, proteins on the lysosome surface can get tagged with ubiquitin. That's like a molecular kick-me sign for degradation. There's even talk of a protein called SPG20 that might act as a kind of triage officer, deciding, repair versus demolition.
Okay, so you repair if you can, destroy if you must. But then you need lysosomes. How does the cell restock its supply after all this?
Right, you need to maintain the pool. That's where lysosome biogenesis comes in. And it happens in a few ways. First, the straightforward way, biosynthesis and delivery. New lysosomal proteins are made in the ER, they get tagged in the Golgi apparatus, and then shipped off to the lysosomes.
Standard protein production line.
Yep. But there's also something called lysosome reformation. New lysosomes can actually sort of bud off from existing ones, especially ones involved in degrading stuff.
Yeah.
It involves processes of tubulation, forming tubes, and scission, cutting them off, so it's not always making brand new ones from scratch.
Like recycling parts of the factory itself. Clever.
Very clever. And crucially, underpinning all this is transcriptional control. There are these master regulator proteins like TFEB and TFE3. When the cell senses lysosomal stress or damage, these guys head to the nucleus. And they turn on the genes needed to make more lysosomal components. It's a feedback loop, basically telling the cell, hey, we need more lysosomes.
So this whole system, repair, remove, replace it, sounds absolutely fundamental. What happens when it breaks down? What are the bigger health implications?
Oh, they're huge. Defects in lysosome homeostasis are linked to a lot of diseases. The most obvious are lysosomal storage diseases, or LSDs. Here, stuff doesn't get broken down properly, it accumulates, and that physically stresses the lysosome. This accumulation can itself make the membrane leaky, contributing to the disease pathology in conditions like Gaucher or Niemann-Pick disease.
Okay, so LSDs are a direct consequence. What else?
Well, leaky lysosomes can trigger autoinflammation. The escaped contents can activate inflammatory pathways, like the NLRP3 inflammasome. This drives chronic inflammation seen in things like atherosclerosis, Alzheimer's, even some autoimmune disorders.
Inflammation. And I think you mentioned neurodegeneration.
Yes, neurotoxicity too.
Yeah.
In diseases like Parkinson's or Alzheimer's, you have these protein aggregates like alpha-synuclein or tau. These aggregates can actually damage lysosomal membranes. This messes up the cell's ability to clear them out, possibly contributing to the spread of these toxic proteins between neurons.
It's a vicious cycle then.
It can be. And finally, there's aging. Over time, lysosomal function tends to decline, partly due to increased oxidative stress. This leads to the buildup of damaged components like lipofuscin, contributing to cellular dysfunction and the decline we see in tissues as we age.
So understanding all this intricate machinery isn't just, basic cell biology. It seems really relevant for potential treatments.
Absolutely. It opens up therapeutic possibilities. As we figure out exactly how these repair and degradation pathways work, well, it raises an important question. How can we use this knowledge? Maybe not just to fix lysosomal problems and diseases where they're dysfunctional, but could we perhaps even selectively destabilize lysosomes in cells we want to get rid of, like cancer cells? Or could we use this knowledge to design better ways to deliver drugs into cells?
Really fascinating possibilities. This has been an incredible look at how adaptable cells are in protecting these vital lysosomes from quick patches to full-scale replacement. Remember to like this video and subscribe. It really does help support the Science experience. Thank you for joining us today.